was my idea, although I just talked to Javad about what people want right now is coffee. And Javad's like, no, what they want is lightning talks. They don't want coffee.
This is switch timing. We have to stay on time.
So we're not having coffee. However, we are close to the finish line. Yes? Oh, yeah, go ahead. Get after it. You do you. We had 60 awesome abstracts come in this year, which was more than we even expected, and they were quite good. So you'll get to see those at the poster session, which is after the break. But 3 rows to the top, these were the only 3 that hit a very high achieving score for us, and we all agreed that they would be great talks. So these are the best of the best. They're going to be quick, but we did build in time. As long as they stick to their 7 minutes, we'll give them a chance for you guys to ask a question of each of those. So store up your questions right now. These will be lightning fast. And then they'll also be downstairs with their posters, so you can continue to chat with them down there.
Okay, so the posters downstairs
after all of this, starting at...
Right after this.
Right after this. Starting it right after this.
Right after this.
One floor down. Thanks, Carl.
Yes,
Sure.
that came from me. Okay, first talk is from Andrea D. Micheli from Zurich. He's going to be talking about unraveling the cellular diversity and spatial architecture of infant-type hemispheric gliomas using multi-omic profiling.
Thank you very much.
Take it from here.
And thank you for sticking around just before coffee. I'm Andrea. I'm a senior scientist and computational biologist at the Children's Hospital. For those who are wondering, this picture has been generated by AI by feeding the title of my talk to a large language model. And in our research lab, we're interested in studying a very rare type of pediatric glioma in babies called infant-type hemispheric gliomas. Like many pediatric brain tumors, these infant-type hemispheric gliomas often arise from genetic dysregulation that occur during development. And at the root of this dysregulation, in the case of infant-type hemispheric gliomas, is this RTK fusion, which restricts some cells from completing their differentiation trajectory and instead forms a tumor. And I want to emphasize in the context of pediatric tumors in general that we don't know when during the gestational period this event occurs, this RTK fusion, and also which type of cell is the culprit here or the cell of origin. And understanding these 2 factors are really key for understanding the disease and maybe have therapeutic implication as well. And this is where single-cell multimodal or multi-omics profiling come in to help us better understand the intratumoral heterogeneity of these tumor entities. And an accepted framework in the field so far is that pediatric brain tumors are composed of these precursor-like cells of fetal or developmental origin that recapitulates the developmental hierarchy, but in a very disorganized fashion. And so when it comes to those infant-type hemispheric gliomas I was talking at the beginning, we don't know their cellular composition, their developmental origins, and how do they compare to other pediatric brain tumor entities. There you go. So since this tumor entity, the infant-type hemispheric glioma, is very rare, we had to partner with many different institutions around the world to collect specimens, including CBTN, and very grateful for this collaboration. And we assembled this unique cohort of 24 cases with RTK fusions, 14 of which are infants under the age of 2. And we conducted extensive molecular analysis on this cohort using single-nucleus, multi-omic, ATAC-seq, RNA sequencing, bulk profiling as well, like methylation, RNA sequencing, whole genome sequencing, and also spatial transcriptomics. Integrating all these patients together, we resolved over 80,000 single nuclei transcriptomes, which we partitioned in those 13 different types of cells, a lot of wild-type cells, but also 75% of cancer cells. Accurately characterize the cancer compartment of this tumor with consensus from the literature, because there's a plethora now of single-cell atlases being developed or published in brain development and pediatric oncology alike. We did this huge integration with 12 different data sets from the developing brain, over 300 signatures. Maybe some of you recognize the studies, landmark studies published on this heat map. And this analysis revealed that these infant-type hemispheric gliomas are composed of 5 different types of cells of both neuronal and glial origins. So you have radioglial-like cells, glioblast or AC-like cells, OPC-like cells, and then neuronal-like cells. And most strikingly, we observe widespread heterogeneity in the cellular composition of these tumors across the patient cohort that we have. And this could suggest, going back to the question of timing at the beginning, that the oncogenic event might occur at different times during gestation, right? Maybe yielding tumors with different composition, cellular composition. However, I want to note that all patients with those RTK fusion have the radioglial-like population, suggesting that maybe this is the cell of origin in those tumor entities. We also did some fancy pseudo-time velocity trajectory model to organize the cellular heterogeneity of this population and inferred a trajectory that really starts from the radioglial-like population all the way to both either a neuronal-like state or a glial OPC or astrocytic-like state. And interestingly, the radioglial-like population also has the lowest RNA velocity score, indicating that these cells are at steady state and may be constituting the pool of cancer-propagating cell, or the cell of origin, in other words. We also have matching full-length transcript single-cell data, so SMART-SIG2, and we could also identify the fusion read in all cell types, right? Further suggesting that radioglial-like cells are the cell of origin. And then finally, when we stratified the cancer cell population across different patient group and age groups, we observed that infant and patient with low-grade histology, as classified by pathology, have a significant enrichment for neuronal-like cells. And that the tumor composition also differs from other pediatric gliomas, like DMGs, H3K27M, that lack the radioglial-like population and have more of the OPC-like. And so neuronal tumors are likely typically less aggressive, and this could explain, and I'm not a neuro-oncologist, why those infants sometimes have a better outcome because of maybe of the greater content of neuronal-like cells present. I also have some spatial transcriptomic data, but I think I'm a bit over time, so feel free to find me at the coffee or cocktail break for further discussion. But just to summarize, we resolved in infant-type hemispheric gliomas this very rare patient population, 5 types of cancer cell population distributed along both the neuronal and the glial developmental lineage, with radioglial-like cells probably being the culprit, the cell of origin, carrying the fusion. In infants, we have more of a bias towards the neuronal lineage compared to older kids. And this maybe could be associated, but we need more data with better survival. And so this concludes my talk. This was really a joint effort of many talented researchers, including Dr. Strukling, the head of my lab, and other scientists in the oncology department at the Kinderspital of Zurich, and as well as our collaborators at Dana-Farber. And I would also like to acknowledge our funding agency, CBTN, for providing samples and patients and their family. Thank you.
Our next speaker is Yiming Deng. Is Yiming here? Yiming, it's a pleasure to have you here. We'll be talking about the high-level expression of SPRY1 indicates elevated MAP kinase pathway activity. Yiming?
Thank you for the introduction. I have no disclosures. Atypical teratoid rhabdoid tumor, or ATRT, is the most common malignant central nervous system tumor of infancy. We're talking about kids less than 1 year of age. Unfortunately, this type of tumor is very fast-growing, and it does not respond well to current treatment options. This means that we are in urgent need for targeted therapy. Previously in our lab, researchers conducted immunohistochemistry on ATRT primary tumor samples. The study discovered that there is a high-level expression of phospho-ERK in these samples. Phospho-ERK is a member of the MAP kinase pathway, and this pointed to the pathway as a potential therapeutic target. We are also actively looking for additional biomarkers for the MAP kinase pathway. Here's one example, which is SPROUTY1. Using the CBTN Pediatric Brain Tumor Atlas database, we revealed that SPROUTY1 mRNA expression is higher in ATRT compared to most other pediatric brain tumors. And this could be potentially something that we work on as an indicator of sensitivity to MAP kinase pathway inhibition. And also previously, our lab worked on a different MAC inhibitor called binimetinib. The study discovered that binimetinib was able to decrease ATRT flank tumor growth. However, unfortunately, it did not really work well in orthotopic xenograft, which is inside the mouse brain. So while we were recovering from the disappointment of this revelation, we also found the silver lining. So since it worked in the flank, it means that the MAP kinase pathway is indeed very likely to be relevant. And to make this work better, we need something that's a lot more blood-brain barrier penetrant. This is where our new drug, mirdametinib, comes in. Mirdametinib is a novel MAC inhibitor that has been reported to have superior blood-brain barrier penetration compared to its predecessors. Here we are using patient-derived cell lines of ATRT to test the efficacy of mirdametinib in vitro. On the left side are Western blots showing decreased phospho-ERK after drug treatment. ERK is the direct downstream effector of MAC, and the phosphorylated version of ERK is the active form. On the right side is a growth curve showing that after mirdametinib treatment at different doses, we can see growth suppression of these cell lines compared to the DMSO control. Moving on, we demonstrated that mirdametinib can also induce apoptosis in these cell lines. On the left side is what we call an Annexin-5 flow cytometry-based assay. The top half of this graph is a typical panel you can see when you conduct an experiment such as this one. The green area in the bottom left is the live cell population, while the red area encompasses both early and late apoptosis. The bottom shows an example using the cell line CHLA06. You can see that after mirdametinib treatment, the cell population shifted from the live cell area a lot more to the apoptotic cell area. This result was confirmed on the right side by immunofluorescence staining for cleaved caspase 3, which also goes by this catchy name that my boss likes calling it by, the executioner caspase. After mirdametinib treatment, we can see a significant increase of percentage of cells positive for cleaved caspase 3, therefore also confirming the same result, that the cells are being more apoptotic. After observing promising data from our in vitro study, we transitioned to in vivo study using immunodeficient mice bearing orthotopic ATRT xenograft. Here is the study using the cell line CHLA06. We had 5 mice per group, 5 for control and 5 for drug treated. Both group following oral treatment of 5 days per week. No toxicity was observed that was really noticeable. On the left side is a Kaplan-Meier curve showing the survival data. We can see that the drug treatment was significantly increasing the survival from 17 days of the control group to 24 days of the mirdametinib treatment group. On the right side is a Western blot analysis using the cancer site of the mouse brain harvested at the end of the survival study. We saw decrease in phospho-ERK after drug treatment. So this was really promising because it meant 2 things. First, the drug indeed got into the brain in the first place. And second, it was able to engage its target pathway inside the brain. And I was really happy when I saw this and thought that was the best Christmas gift I got after doing months of mouse work. A challenging but also exciting thing about cancer research is that a single agent treatment is rarely sufficient to eradicate cancer. Therefore, we are seeking combination therapy options right now to enhance the treatment efficacy. Specifically here, we're exploring the possibility of combining MAP kinase pathway inhibition with mTOR pathway inhibition in order to achieve better outcomes. I think a previous presentation kind of alluded to it, but the MAP kinase pathway and the mTOR pathway are somewhat sister pathways, both starting from similar receptors and then both leading to things like cell survival, cell motility, and cell proliferation. So here we use 2 drugs, mirdametinib, which is the MAC inhibitor that I just talked about, and paxilosib, which is the PI3 kinase inhibitor. capable of inhibiting the mTOR pathway. On the top part of this page, we can see growth curve showing, again, suppression of growth, but much more from the combination treatment than the single agent. And the bottom is the apoptosis data. This time we're probing for cleaved-PARP, which is another apoptosis indicator. There's a lot more cleaved-PARP for the combination condition compared to the single agent, therefore also showing that there's enhanced efficacy. In conclusion, ATRT expresses high level of Sprouty-1 mRNA, and it also has high level of phospho-ERK, therefore demonstrating upregulated MAP kinase pathway. And second, mirdametinib has good blood-brain barrier penetration and was able to achieve survival extension in vivo. Moving forward, we are trying to see if we can get more from the Sprouty-1 side of the story to see if it can be used as a biomarker for predicting susceptibility to MAP kinase pathway inhibition. And we are trying to test and explore combination therapy options to further enhance our treatment efficacy, especially in vivo. And I'd like to thank everybody from our lab who has helped me a lot along the way, and also our funding sources that has made all of this possible. Thank you.
I was going to ask how you think the suppression of MEK gets you to apoptosis.
Yes, so at first we thought that maybe since the MAP kinase pathway is more proliferation related, it would be more of a cytostatic effect. But we did discover that the apoptotic effect is also super noticeable. The exact mechanism is really what we're trying to get from this study. So we're actively trying to decipher the mechanism of that. Apoptosis is programmed cell death. So if you subject the cells to something like external or internal stress, the cell has a mechanism built into its genetics and its pathways that it will kill itself. For normal body cells, this is a protective mechanism to prevent bad things such as cancer happen. Cancer cells also, a lot of them retain this pathway, so we can use it as a way to kill cancer cells as well.
Okay, last but not least, last talk will be Elton VanNoy from Children's National. Talk titled Manufacturing CAR T Cells with DNA Methylation Inhibitors Enhances Their Fitness in Immunocompetent Solid and Brain Tumor Models.
Good afternoon. Hi, everyone. I'm Elton VanNoy from Children's National. I'm a second-year PhD student in Dahlia Hadar's lab, and today I'll be discussing how we can enhance CAR T-cell therapy by using DNMTI treatment during manufacturing against solid tumors and brain tumors. I have no disclosures. So we'll be discussing chimeric antigen receptor therapies, or CAR T-cell therapies. And this is where you design a chimeric antigen receptor that is specific for tumor-associated antigens. You can profile brain tumors to find the most frequent occurring surface antigens and design CARs specifically to target those. So they are highly targeted, and as you can see, they will bind to the tumor-associated antigens, specifically avoiding our own cells, and then inducing cytokine release and cancer cell death. But when we're targeting solid tumors, we face some unique challenges, specifically as antigen exposure, chronic antigen exposure will induce exhaustion in this T cell, leading to a lack of persistence, terminal differentiation, and ultimately the inability to eliminate the cancer. So we want to investigate how we can curb this CAR T-cell exhaustion and have them last longer to be persistent against these solid tumors in the brain. And this is where we turn to epigenetics. And this is mostly regulated by DNA methylation, demethylation of promoter sequences that are important for important effector genes. When T cells are exhausted, we see more of this methylation in the cell. So this is where we are looking at DNMTIs, or DNA methyltransferases inhibitors. And these are compounds that block the methyltransferase and prevents these promoter genes from being methylated, allowing these effector genes to be expressed long term. We are hopeful that by using these DNMTIs during the treatment and manufacturing of our CAR T cells, we can prevent the methylation of these promoter genes and have these effector genes being activated, preventing exhaustion. And it's really important that we're doing this during the manufacturing phase of our CAR T cells because we're enabling another therapeutic, but at no cost or additional treatment to the patient. So this workup to make these DNMTI treated CAR T cells starts with the isolation and activation of murine CAR T cells, as we are starting this work in vitro in a murine model. And then from there, we do the retroviral transduction of our CAR. We'll be using a B7H3 CAR, which has been profiled on pediatric brain tumors. And we also have our control CAR as well. During this transduction period, we are doing low dose treatment of DNMTIs. We have selected a panel we are investigating. For today's presentation, I'll be showing 5-azacitidine as our first DNMTI of interest. And then from there, we have a handful of functional assays that we can use to quantify how well these CAR T cells are persisting with this treatment. And since we are doing the transduction the same time as drug treatment, we wanted to note that this does not affect the transduction efficiency of the CAR. So we're good to go forward. The first assay I want to discuss is our tumor cell viability assay, where we co-culture tumor cells and CAR T cells at a decreasing effector to target ratio to see and quantify how cytotoxic these CAR T cells are. We'll be using GL261, which is a murine glioma, for these assays. Here we can see that when we co-culture these together, our control CARs, as expected, don't cause any cytotoxicity. But as expected, with our B7H3 CARs, at higher concentrations, we're seeing more tumor cell death, and it tapers off as we go into our lower concentrations. This is our first stimulation, so the very first time our CAR T cells are interacting with tumor cells. So this is a nice dose-dependent curve that we expect to see. Although we are seeing a little bit of differential activity between our different DNMTI groups, at this stage we don't expect to see much difference, as again, this is not persisting very long yet, but it is exciting to see, and this will encourage future studies, as you'll see. What we really want to talk about is persistence. We use our signature assay, repeat stimulation, where we co-culture T cells and tumor cells. We let those intermingle for 3 days, and then we collect our T cells, count them, and replate them with fresh tumor cells. And we do this as many times as possible until the T cells stop expanding or they stop killing. And this is how we quantify our long-term persistence for our T cells. And here we can see, again, our CAR T cells don't expand, don't persist, don't kill. But then we see our non-treated B7H3 CAR T cells. They are killing the tumors and they are persisting, but they aren't expanding very well. Compare this to our DNMTI-treated CAR T cells. We are seeing a massive increase in proliferation from our 150 nanomolar treated CAR T cells. Another way to look at this is that we can see our 50 and 100 nanomolar treated CAR T cells are living for up to 13 repeat stimulations, which is almost 2 months in vitro. And then we can also see that they are expanding significantly higher for fold expansion compared to our 0 and 200 nanomolar. So this is really exciting to see as an avenue to persist these CAR T cells against solid tumors and chronic antigen exposure. Additionally, this is some supporting evidence with that tumor cell viability assay I showed at the 4th stimulation. So later on, the longer these CAR T cells are persisting, here's a snapshot of specific concentrations. We can see the longer these CAR T cells are persisting, they're actually more cytotoxic in those ideal concentration range at 1:1 and lower concentrations like 0.25. To further support this, I threw in some preliminary data we're working on now with a new DNMTI GSK that we're looking at. And here we can see that even down to the 8th stimulation, we're not only seeing that differential cytotoxicity between the drug treatments, we're seeing increased cytotoxicity. So not only is the DNMTIs allowing the enhanced persistence of these CAR T cells, it may be enhancing cytotoxicity over time as well, which is really exciting. So to wrap everything up, we're seeing some really interesting data that 50 to 100 nanomolar improves the proliferation of B7H3 CAR T cells, and that this may also be enhancing persistence and cytotoxicity long term. We are currently employing different DNMTIs, CAR constructs, and also looking into other solid tumor models to expand this work and hopefully find some interesting data. We are hopefully going to move in vivo very soon and hopefully have some new data in the future. So big shout out to my team at Children's National. Like I said, I'm a newer PhD student and it's been a fantastic project to work on and I'm excited to keep going. So thank you. With some of the DMTIs, specifically AZA, we're seeing at higher concentrations, it just completely wipes the cell out, even earlier than non-treated. And this is probably just due to an aberrant decrease of that methylation, because with exhaustion, you are seeing more methylation than usual for those important promoter genes. But if you're completely inhibiting that methylation, there's some protective housekeeping genes that are getting affected as well. So that's probably finding that sweet spot of altering epigenetics where you're kind of helping the cell out, not getting exhausted, but not getting into other territories as well.